Hello, biology students. We're working on the Concord Consortium Connected Bio Deer Mouse Fur Color module. We're on Activity 5, Lesson 2.2. We're on page 1, and we're investigating the light brown fur color. Remember, we have two types of mice that we're studying. One is from the field, and remember, the field environment looks like this. So these field mice tend to be darker. And then we have a beach environment. And so the mice, here's the field environment. Mice in this field environment tend to be darker. And then we have this beach environment where they tend to be lighter. And so we have two mice that we're studying here. We have the beach deer mouse that lives in this environment. And we have the field mouse that lives in a much darker environment, more like the woods, and matches in with soil and tree bark and things like that, where the beach deer mouse matches the light sand. So we're at the population level here in our multi-level simulator, and we want to get to the organism. So we're going to collect a couple mice. So they'll just magically appear in the box over there when we collect them. Then we can be done collecting and click organism here. We've dragged a mouse here so we can see at the cellular level, these are the cells at the base of the hair shaft that provide the pigment to make different colors of hair on different mice. We are also going to measure different substances in different locations on this mouse. Let's put the same mouse here. And let's zoom in to the skin level, to that single cell level. And then let's zoom in again. So we're at the cell membrane level and we can see a little bit better. So this is the same mouse and this is two different views. This is a larger kind of view from further back. Less magnification. This is more magnification. We're looking at a part of the cell membrane here. So we're blowing up like this part of the cell to look at right here. So we're going to measure 4 different substances, the hormone, the activated protein, the pheomelanin, and the eumelanin in our 3 different locations, the extracellular space, the cytoplasm, and a melanosome itself. Now let's see if we can get it. There we go. Melanosome. So let's start with the hormone, and we're going to just measure, not add. So let's measure in the extracellular space. Is there any hormone going to be out here? Well, it looks like in our graph, yep, some hormone is in the extracellular space. Is there any in the cytoplasm? Nope. Is there any in a melanosome? Nope. And that makes sense. The only place that the hormone is active is in the extracellular space, so that's probably the only place that we're going to find it. Let's see if there's any eumelanin here. Remember, that's the darker of the two pigments we found in the dark brown field mice. So let's measure the amount of eumelanin. Is there any in the extracellular space? No, that makes sense. That's not really where it's supposed to be. How about the cytoplasm? Nope. How about one of these melanosomes? Now we would expect that there would be some pigment in a melanosome since that's where pigment is made, but there's not any of the eumelanin here in this particular mouse. Let's see if there's pheomelanin. Or pheomelanin, excuse me. Pheomelanin is the light brown pigment. Let's measure it in the extracellular space. Nope, doesn't show up in the graph here. Let's measure it in the cytoplasm. Nope, doesn't show on the graph here. Let's measure it in a melanosome because we would expect it would be there, and it does show up here on the graph. I'd measured it before, so that bar was there from before. And now let's measure the activated protein here, activated protein complex. Is it in the extracellular space? Nope. Is it in the cytoplasm? That's where it should be, because that's where we saw it the last time we measured this in the darker cells in the field deer mice, but we're not seeing it here. Let's try to measure it again in the cytoplasm. Nope. And that's unusual, and that's something we should pay attention to. Is it in the melanosome? Nope, but we wouldn't expect it to be there because that's not where it's active. So really the only two things that we can measure out of these four we had in the last cell are the hormone and the pheomelanin right here. So we're going to want to fill out a table here, and I have filled out this table for you just to make it easier. I know it's really confusing with all these different substances and finding them in all the different spaces. So I went through and I said, did we find pheomelanin anywhere? And no for the extracellular space, no for the cytoplasm, and yes for the melanosome. Activated signal protein we didn't find anywhere. Eumelanin we didn't find anywhere. And hormone we only found in the extracellular space and not in the cytoplasm or the melanosome. So if that helps you organize your thoughts, I hope that's helpful. Let's go back and do some more investigating. So we measured the 3 locations, we did our field notebook, table 1. We're ready to go to the next page here. So now we need to make some predictions using the data we've collected so far. Using the table and the measurements, predict how the fur color would change in the light brown mouse when you add each of the different substances. So instead of just clicking on the measure, we're going to click add. And we're going to try all these different substances. But before we do that, we're going to try to predict what's going to happen. And you might want to refer back to some of the data you collected in lesson 2.1 to think about this. It's interesting that there's no activated signal protein. There's no eumelanin here. But what do you think is going to happen when we add eumelanin? Or what do you think is going to happen when we add pheomelanin? Will the cell get lighter when we add pheomelanin? It's already pretty light. Will the cell get darker if we add eumelanin? How about the hormone? Is that going to work? It doesn't seem like there's any activated protein. So the hormone may not work in this type of cell. So you can do some predicting there. Stop the video, and when you're ready, we'll go on here. So we're going to add substances to 3 locations in the melanocyte. Record the cell if it gets D for darker, L for lighter, or nothing changes. Repeat the process several times in each location to be sure you know exactly what's going to happen, or you know exactly what is happening, and you just didn't have a fluke. So let's go full screen here, and we'll see what happens when we add hormone here. Did it get darker? No. Let's try it again. Nope. How about the cytoplasm? We'll add it in the cytoplasm. Nope. We'll try adding it up here on the larger view. We'll add it to melanosome. And a little bit of hormone appeared in the melanosome, but it didn't do anything. The cell didn't change. All right, let's try eumelanin here and see what happens. So I'm going to add eumelanin to the extracellular space, and it appears there, but it doesn't change the color of the cell. I'm going to add eumelanin to the cytoplasm. It does appear on the graph there, but it doesn't change the color of the cell. If I add it here in the melanosome, the cell gets darker for a bit, but then it lightens up right away. So eumelanin does work to darken the cell, but I don't know that the cell can produce any of that. Let's try pheomelanin. We can add it in this extracellular space. Nope. We can add it in the cytoplasm. It shows up, but it doesn't do anything, doesn't change the color. And we can add it in the melanosome. It shows up, but it doesn't change the color, suggesting perhaps already that pheomelanin is the dominant melanin here in this cell, and that there is no eumelanin unless you specifically add it, which only happens in our case because we're experimenting. The cell doesn't actually have eumelanin itself. Let's add activated protein and see what happens. We can add it in the extracellular space. It appears, but it doesn't actually do anything. We can add it in the cytoplasm where it usually works, and there it darkens up the cell a little bit, but then it disappears. And then in the melanosome, it doesn't really do anything. So if we add activated protein, it works. If we add eumelanin, something happens. But nothing happens with the pheomelanin and the hormone. And those are the only things that we find, that we measured in the actual cell. We can add activated protein, we can add eumelanin, but they don't actually appear in this cell when we measure it. And you can see for the activated protein, you can see what's going on down here. You can see that the protein complex, those blue globs that are together, they don't separate and go into the cytoplasm like they did in the darker cell. We'll see the darker cell here in a moment. And we see the melanosomes, but we don't see the melanosomes getting darker here. We see them stay this pretty light color. We see the hormone landing, but we don't see that hormone telling the receptor to split this activated protein like we did in the darker cell. We don't see this blue comma kind of split off and go by itself into the cytoplasm and signal the cell. So, and we did, hopefully you made some predictions. They might have looked like this. These are actually our measurements as well. So for pheomelanin, we didn't see any changes. For activated signal protein, we saw it get a little bit darker. For eumelanin, we saw it get a little bit darker in the cytoplasm, or sorry, in the melanosome. That's incorrect, excuse me. And the hormone, we didn't really see it work anywhere. So hopefully your predictions matched our measurements. I'm actually just going to copy this down because these are our measurements. So the next thing we'll do is we'll be heading to Table 4 as a summary of what we learned. We saw color changes when the substance is added, and we have to come up with a new model because this table looks very different than it did when we were working with the darker cells, the field mice cells that had much darker fur. The hormone worked in the extracellular space, we'd see a change, and in the darker cell we saw pheomelanin make a change when we put it in the melanosome. And so there's some differences here. This cell may have some mutations or some changes in some of the proteins in the receptor, maybe the hormone, maybe the activated signal protein is different. That's causing the cell to not have eumelanin and not have any activated signal protein that we can measure. Let's remind ourselves of the differences between this beach mouse cell and the cell with more coloring. A dark brown mouse from the field here let's zoom in here and zoom in here and just see if we can compare them, and then we can maybe build a better model of how different the light cells are from the dark cells, and how those light cells, the model for how the light cells signal to make melanin, because it's obviously different than the dark cells. Here in the dark cells, if you watch what happens, the protein complex comes up to this receptor, and when the receptor gets a hormone, it breaks up the protein complex. So you can see the protein complex come in, and it doesn't break up until there's a hormone here. So we have to wait. Up, as soon as a hormone comes, it breaks up, and that comma-looking protein, the comma-looking protein breaks off and heads into the cytoplasm. And that signals the cell. If we put in activated protein, if we add that to the cytoplasm here, that makes the cell darker. That doesn't work in the light cell. Only the pheomelanin works in the light cell. Or pheomelanin, excuse me. If we add some eumelanin to the melanosomes here, it makes the cell darker. Let's add this more clearly. There you go. So if you add eumelanin, the darker pigment, it makes the cell much darker if you add it to a melanosome in this dark cell. But that doesn't happen, that the light cell isn't making that eumelanin. How about the pheomelanin? If you add that to the melanosome, it makes this cell light. It doesn't make the other cell lighter. It actually makes it just a skosh darker. The light cells get just a little tiny bit browner when you add the pheomelanin. So if we want to make some predictions here, let's go back to our other page here, where we're supposed to be building a model. Show how the substances affect the cell and produce the light brown fur color of the light brown mouse. In our previous model looked like this. There's a hormone that activates the receptor to break up the activated signal complex protein here. The half of the activated signal complex goes into the cytoplasm, which tells the cell to make melanin in the melanosome. But in this light cell, we don't have any activated signal protein. We're not making eumelanin in this melanosome. So something is going on here, something between the hormone and the receptor and the signal complex. This is not working the same as it did in the dark cell. So this part, something is different. There's no activated protein that's going to tell the melanosomes to make eumelanin. So we must, the cell is only making pheomelanin, which is very different than eumelanin. It gives the cell a different color and hence gives the fur a very different color. So we have to come up with a new model. This is the old model for the dark brown cells of the field mice. We have to come up with a model for beach mice. What we do know is that we have some kind of melanosome making pheomelanin, and when we have pheomelanin, the cell does get a little bit browner. We don't know the mechanism in this lighter cell from the beach mice. What is cueing that pheomelanin to be made? So we have to come up with some kind of new model as we study these beach mice. And in the next lesson, lesson 3.1 and 3.2, we'll be looking at the proteins that are produced in these two different systems. So we'll be refining this model here. And by the way, you can't draw in the program in Concord themselves. I'm using a program called IPEVO to draw on my screen here so you can see it. Make your own drawing on a piece of paper in an art app, and then you can upload that image, either take a picture of it or make a JPEG, upload it, and that can be part of your lab report. So make your own model and upload it. And we came here and we measured in the 3 locations. Well, we'll measure just to be sure. Let's measure in the cytoplasm. We're measuring hormone. Yep, we found it in the cytoplasm. Did we find it in the extracellular space? I guess you measure everything at once. So we find activated signal protein in the cytoplasm in this model. We find hormone in the extracellular space. We find both pigments in the dark brown cell in melanosomes. This is different than in the beach mice. So I recorded my observations. There's no eumelanin, there's no dark pigment. The blue protein complex is not in the beach mice. I should preface this. In the beach mice that are lighter, no eumelanin is made, no dark, so there's no dark pigment. The blue protein complex in the lighter is not broken up like this one. No signal goes to the cytoplasm telling the melanosomes to make the melanin. When we measured it in the lighter cell, there was no activated signal complex at all. And there is here in this darker cytoplasm. So record your observations from investigation 2, comparing the mice at the cell membrane level. Refer to the key below to identify the proteins and substances inside the cell. This is the same key you made in lesson 2.1. So here in the, let's go to this lighter mouse here. Let's look at a lighter mouse. Let's put this mouse back. Oh, it's not letting us do a lighter mouse. Well, in the lighter mouse, the signal protein is not activated by the receptor protein. If there's no activated signal protein, the melanosomes don't or do not make any eumelanin. And so the cell is lighter. We're talking about the beach mice, not the field mice, which are illustrated here. So based on your understanding of the simulation and the models we've made, make a claim about the difference between the melanocyte cells in the dark brown mice and the light brown mice. So we ask some questions here. In class, you may want to make a claim about the light mice can't make eumelanin, or they can make eumelanin, or pheomelanin is the most important melanin in the beach mice. And then explain how the difference causes the mice. So make a claim about the types of melanin or what's causing the differences, and then explain how the mice have different fur colors. I'm just going to give you some questions you can think about answering in this question number 7. So, is pheomelanin making the mice lighter on the beach? They don't produce eumelanin, which is dark pigment. So is pheomelanin, the lighter pigment, the only thing coloring their fur? So question number 8, I'm going to let you draw a picture of the structural differences in the proteins of the 2 mice. Describe how these differences affect how the proteins function. So here's the activated signal protein. In the dark brown mice, it does break up. That blue comma breaks off. If we go back to the light mice and we go full screen, watch the activated signal protein come to the purple receptor here. Even if it has a hormone docked, that activated signal protein doesn't break up. There's no activated signal protein, there's no blue comma protein that gets into the cytoplasm and tells the melanosomes to make eumelanin, that darker pigment. So it's probably, when you draw your model, it's probably something to do with this activated signal protein that doesn't get activated by the receptor and the hormone. There's something about the way that the activated signal protein and the receptor and the hormone all link up that isn't breaking these 2 proteins apart, the 2 blue proteins apart, and getting that blue comma to go into the cytoplasm and tell the melanosomes what to do. So now's a great time to figure out what you did and what you learned in this part of the lesson. They've filled in what you did here. You added specific substances to different locations of the cell to see the effect in the light brown mouse. What differences did you find? What did you figure out? How does this answer some of our driving questions about what causes those differences between the field mice and the beach mice? We're really trying to figure out why the field mice and the beach mice are different colors. And not just because hawks eat one or the other, but what's happening at the cellular level to make those fur colors really different. Once you've filled out your table and you've made your model on the previous page, you can answer question 11. How do you think the difference in the proteins relates to the fur color of each mouse? You can talk about pheomelanin. You can talk about eumelanin, the dark protein. You can think about the activated signal complex and how it does or does not get activated. And the hormone and receptor complex. How is that involved? For question 12, tell me what differences or what questions we still have about how different fur color is produced in the dark brown field mice versus the light brown beach mice. Write your questions here, and you can also put them on the driving question board. The driving question board looks like this, and you have the link to it in your tables document, so you can check out that driving question board and you can add to it. Thanks so much for watching. Make sure you click Show All Answers. You go to the report page, and on the report page, you find that little square with the up arrow. Click that. You'll find a menu that says Options at the very top in tiny blue letters. Click Options, then click PDF. Back out of the menu, click this little square again, and then click Notability, and you can download your answers and put them in Schoology. Thanks so much for watching. I'll see you next time.